So welcome everybody. Thank you so much for joining. This is, wow, I don't know which number minor symposium this is, but this is our fall 2020 minor symposium. And just as a recap, so this semester we have 7 students who have completed research and are scheduled to share their work with us. And as a recap of what they have accomplished so far, in addition to these independent research projects, every minor student has to complete 7 required credit hours. That's a general ecology course and the applied ecology course. And then they have to complete a minimum of 2 electives for a minimum of 6 credits. So that's 13 credits that are prescribed. And then in addition, a minimum of 3 credit hours. So that's 135 contact hours, no small feat, helping to ask a unique, novel scientific question, gather the data necessary to address that question, and then analyzing those data, working closely with a faculty mentor or with a mentor outside of academia. In these cases, we have a mix of different mentors from different arenas whose students and projects will be represented today. So that's our rundown of all the accomplishments that culminate in these presentations for the Applied Ecology Minor Research Symposium. So thanks for joining us. I'm really excited about the diversity of projects and perspectives. And I do want to start off with Jared Lamb. So Jared, a senior in fisheries, wildlife, and conservation biology. You're going to get us started, all right?
Thank you. Let's see if I can get this screen shared correctly. Okay, so as she said, my name is Jared Lamb, and I'm a senior in fisheries, wildlife, and conservation biology. And my project my research was, how do butterflies respond to forest type transitions? And I conducted this research in the summer of 2019. And it's a pilot study of pollinator response to restoration efforts of a rare longleaf pine ecosystem. And I conducted this research through the Dorstu Conservation Scholars Program. And I also presented it at the 2020 Ecological Society of America conference that was virtual this year. And some background on this research. Over the past 200 years, longleaf pine ecosystems have drastically decreased along the southeast. And so these have been replaced with the loblolly pines. And so there's been a cause for restoration and conservation of these ecosystems. And so this brings up a topic of Piedmont longleaf pine ecosystems in North Carolina. And actually around Raleigh, there is a rare longleaf pine ecosystem, and that is classified as the dry Piedmont longleaf pine forest. And these are determined by soil factors and unique environmental characteristics that set these apart from different forest types. And NC State actually has a biological field lab on Lake Wheeler Road, and it's used with many natural resources and College of Agriculture and Life Science classes. It's called the Lake Wheeler Road Biological Field Laboratory. And it was actually discovered that this field lab was historically one of these dry Piedmont longleaf pine forest. And so there's been a restoration project of this forest type at this site since, and it began in 2018. So some more background, this restoration effort presented the opportunity for a long-term monitoring study on how the ecosystem responded to this restoration over time. And this allowed for documentation to be collected and progress to be quantified. And so biodiversity measures would be important to measure the success of this restoration effort, because longleaf pine forests are typically known as biodiversity hotspots. And so the image to the right is kind of a description of how this biodiversity hotspot dynamic works in this longleaf pine ecosystem. And you can see there's many different layers, which allows for biodiversity. And this picture actually does a pretty good job of displaying the structure that would occur in a mature dry Piedmont longleaf pine forest. And also a key component to a healthy ecosystem is its pollinators. And the pollinators will allow all these multiple vegetation types and structures to occur. And with the decrease in pollinator populations across the globe, as we have noted, monitoring the response of these pollinators to this restoration effort is a key component. And so I chose for this project to monitor Lepidoptera, which are butterflies, as they're easily identifiable and one of the more easier pollinators to survey. So my typical general research question going into this project is, how will Lepidoptera respond over time to the forest type transitions within the site? And the questions I answered within this study was the composition of butterfly species throughout the site, and does species composition vary by canopy cover? And so the answer to these questions provides a baseline for future studies at the site, and it can provide further knowledge research going forward. So my method going into this project, I set up 3 10 by 10 meter plots to survey, and these plots were selected based on low, medium, and high canopy densities. And you can see the general density of these plots by the shade on the ground. And these were, I measured these plots densities by using a spherical densiometer, and I set up in each plot, I set up 4 quadrants with 5 different points, measuring points in each quadrant and in the middle of the plot. And you can see this in the image in the top right in this slide right here. And you can also see a map of where I selected where these plots would be. These are the locations of each of the plots. And so the image in the top right is how I measured canopy density. I used a spherical densiometer, and I averaged all 5 of these points in each plot. And so a common butterfly surveying technique is called a pollard walk. And this is basically just walking a transect every day, multiple times a day at peak temperature times to look for butterflies and identify them. And so in the bottom right, the black lines show the transects that I walked, and the red lines show the observational buffer in which I limited myself to look at for butterflies in each transect. And so I took data during 6 different observational periods, and this occurred between July 28th and August 30th. So about a 1 month period in the middle of the summer, which allowed me to take these surveys in ideal temperature times for butterflies to be active. And I also used a super zoom camera to identify the butterfly species, as it allowed me to get fine details without spooking the butterfly off the plant that I was looking at. And this was important to me too, because I was kind of new to butterfly identification, and I couldn't really do it based on sight at the beginning. So allowing to take pictures and look at them later using a field guide really helped me identify the species. And so results, this was my first question: what is the composition of butterfly species throughout the site? And I observed 8 different species. Most of these pictures are mine, but some of them I had to get off the internet because some of the butterflies would not stay still for me to take a picture of. And so I observed the common buckeye, the cloudless sulfur, Carolina satyr, eastern tiger swallowtail, gem satyr, gray hairstreak, red banded hairstreak, and the red spotted purple. And so for my second question, does species composition vary by canopy cover? I did the preliminary results of this non-metric multi-dimensional scaling analysis did show that the butterfly species varied depending on the canopy cover of the site. And so as you can see, as we go to the top right of this scaling analysis, that means the percent openness increases. So the more canopy openness, which site 3 exhibited, you get common buckeye and cloudless sulfur. And then in the medium density ones, which is site 2, you get more of the hair streaks, red banded hair streak, and the gray hair streak. And at site 2, which was the high canopy density, low canopy openness site, you get more satyrs, red spotted purples, and eastern tiger swallowtails. So in conclusion, this preliminary study shows promise for the opportunity of expansive biodiversity across the site as the restoration of the dry Piedmont longleaf pine forest progresses. And as a pilot study, this was designated to gain an initial knowledge of the species composition of the site and patterns of presence of butterflies along different canopy covers. And so in the future, more extensive measurements such as temperature, vegetation surveys, and more expensive plots would create a detailed analysis of butterfly species throughout the site, and it gives a basis for further research as the site develops along its restoration progress. And so I just want to give a special thank you to Dr. Leggett. She is the director of the Dorstu Conservation Scholarship Program at NC State, and she helped me a lot in kind of narrowing down what I want to do as a project. And I also want to thank Dr. Jody Forrester and Jacqueline Housley. These are the two, Dr. Jody Forrester was the faculty member that I worked under, and Jacqueline Housley was the graduate student that I worked under. And they helped me a lot with this project, kind of ironing out the kinks and helping me develop the scientific process, as this was kind of new to me. And of course, I want to thank the Dorstu Conservation Scholarships Program to allow me to do this research, and the Applied Ecology Program to allow me to present this research. And if you guys have any questions, I will do my best to answer them. And thank you all so much for being here.
Awesome. Thanks, Jared. Hopefully you're seeing some of our participants are using their reaction emoticons to applaud you. Feel free also to unmute and give a yippee if you want, or just unmute. And do we have any questions for Jared? We have a few minutes for questions. You can feel free to type those into the chat or just unmute and ask them.
I have a question. Jared,
Yeah.
that was a really awesome talk. I was wondering, I don't know much about longleaf pine, and I was wondering if you notice that like plant composition varied across your sites with the different openness. I'm sure those butterfly species tend to hang out like with different host plant species. Did you notice any differences while you were surveying?
Yeah, there was a big difference. So across the site, it's relatively new in the longleaf pine like restoration progress. So there's mainly just seedlings of longleaf pine at this site. But that's one of the main things that I really wanted to do, but didn't have the time to do with this project, was do vegetation surveys and figure out, because butterflies respond to the vegetation types across the site rather than just canopy openness. And so there was a difference in plants between the medium canopy densities and like the high canopy density sites. There's a lot of fireweed across the site because the site has also recently been burned. So yes, there was a response to the vegetation types across the site, and hopefully future studies can kind of survey how these butterflies respond to the species of plants rather than just the canopy density, or on top of the canopy density as well.
Cool. Thank you.
Yep. Thank you.
And I think we have time for one more question if anybody else has a question for Jared.
All right, well, thank you all so much for coming, and thank you, Dr. McKinney, for all this.
Yeah, absolutely. Thanks very much, Jared. Up next, we have Sophia Uthe, a senior in environmental technology and management.
Hi, everyone. Let me share my screen. Let's see. All right, can you all see this?
Yep, perfect.
Awesome. Well, starting this past summer, I was working, I was working at CMAS, which is NC State's marine science facility up in Moorhead, North Carolina, and I was exploring the microbiome associated with eastern oysters. So this summer, 90% of cultured oysters have died, which is a major problem considering that oysters are filter feeders. They clean our water for us. They provide profit through the industry of it. They provide jobs, food, more fish. And the reason for all of these cultured oysters dying is most likely due to a combination of factors, not just one. So the first obvious one being environmental stressors. North Carolina gets a lot of hurricanes, and those rainfall events bring in pesticide runoff and causes fluctuations in temperature and salinity. And that could definitely be one of the factors of why these cultured oysters are dying. The second option to think about would be selection of traits in cultured oysters. Most cultured oysters across the entire eastern United States are triploids now, meaning that they have an extra chromosome and they're sterile. So it's possible that selecting for our oysters that we're growing to be sterile and grow faster, grow longer, they could be more susceptible to stress, which could cause them to die. And the third option could be a possible change in the microbiome within the oysters. So what exactly is a microbiome? A microbiome is a community of microorganisms that are in an environment. In this case, the environment is the oyster itself. And these microbes could be bacteria, viruses, fungi, but what we take a look at the most is bacteria. So if you can see in these little diagrams, in the oyster, the oxygen and all the water, suspended particles, and all the microbes go down first through the gill, and then they go to the stomach and then leave as clean water. So with the gill being the first organ of the oyster that has direct contact with the environment, they accumulate a lot more microbes than any other organ in the oyster's body. So that's why the gill is the best organ for us to analyze for changes in the microbiome. On the other hand of this project, we wanted to take a look at metabolomics. So metabolites are small molecules such as amino acids or sugars, and they're produced as a result of genetic coding specific to a cell or organism or as a response to an environmental stress. So being able to look at the metabolites or the full spectrum of metabolites in an organism at once, we're able to give a snapshot of the oyster's health at that exact specific time of sampling. In other words, looking at the metabolites shows you what the oyster is putting its energy into, like whatever it's responding to, whatever stress it's putting energy into creating this metabolite to survive, basically. So the main objective of this project is to better understand the ambient microbial community associated with cultured oysters and how exactly that microbial community changes over time in correlation with these summer mortality events. Our hypothesis is that a change in the microbial community associated with cultured oysters does occur during a mortality event. And secondarily, we wanted to take a look at the differences in how these oysters are expending their energy using metabolomics. So we only have 3 sites currently because we are sampling opportunistically, meaning that the oyster farmers have to want to work with us and have to want to voluntarily give us some of their oysters. And yeah, they're not too happy about the state pathologists coming by their farms right now, but hopefully if more people get involved, then we will get more sampling sites in the future. But as of right now, in the summer, we had a site at Salters Creek in Nelson Bay, in Bogue Sound near the CMAS facility, and in New River, Jacksonville. To begin with, we would go to the oyster farms, we would collect oysters and water samples, clean the oysters, shuck the oysters, go up to the lab, and then we'd run the water through chlorophyll filtration. And then with the shucked oysters, we would take a gill and a cross-section sample. So if you look at this third picture right here, the little winged part is the gill. We would cut samples from that, and then the cross-section would contain pieces of the gill, the stomach, the gonad, basically every organ we would want to take a look at under a microscope. So from there, we would take that cross-section, we would put that in a cassette tape, the green little squares thing down there, and we would send it off for histopathology that we would then look at later to either say, was this oyster healthy or unhealthy based on gill erosion? The water and gill samples, we would just put in 5 milliliter tubes and stick it in a freezer for later. For the first part of this project, trying to look at the bacteria composition, we wanted to isolate the DNA from the gill and water samples. And we used a technique called magnetic genomic bead DNA isolation. which is where you put the Gill sample in the little tubes, add this magnetic bead, and then stick it to a magnet, basically allowing you to clean your sample repetitively until all you have left is pure DNA, which we then quantified using light spectroscopy, and we sent off to UNC Chapel Hill for them to give us back the 16S gene sequencing of our data. And the 16S gene is really cool because it gives you a really broad overview of all the bacteria present. It can't tell you the species, but it can tell you the genus at least. For the metabolites, we did metabolite extraction, ran that through a nuclear magnetic resonance magnet, which you can see in like the back of that picture there. It's vertical and has a very, very tiny hole, so really small samples can only go in that. So it's not like an MRI meant for humans kind of thing, but same principles. And from that, we would be able to get a spectra of the metabolome, which I'll explain later. But from that, we're hoping to get a workflow of how to use packages in R to be able to analyze all of the spectra data that we get from it. So here is the principal component analysis of our bacteria that we sent off to Chapel Hill. And as we can see that I circled over there, those are the unhealthy oysters, and all the triangles are the healthy oysters. So this tells us right away from the start that there is definitely a difference in the microbial community between healthy and unhealthy cultured oysters. Here's the relative abundance of the top 10 bacteria present that we found, with the first column being our control, Bogue Sound, near CMAS facility, then New River and Salter Creek. And for our control, all we saw was like very common estuarine bacteria that we would expect. And the fecal indicating bacteria we're not too worried about because the sampling event was right after a big like rainstorm, and it's really close to like downtown Morehead City. So it's just like, that's nothing to be too concerned about. But in the New River and Salter Creek, interesting enough, we found bacteria that is anaerobic, meaning that it does well with a lack of oxygen. So that mixed with the bottom row, you see the Vibrio, those are facultative pathogens, which means they're secondary pathogens and they are opportunistic. So what this looks like right now, what we can infer from this is that if there's anaerobic bacteria present, that means maybe the stress is there's a lack of oxygen in the water, which allows opportunistic bacteria to come in for the kill, kind of. To break it down further about the opportunistic Vibrio, we wanted to see if this was one species that was like working against the oysters or if it was multiple. So from this phylogenetic relationship, we see that the Bogue Sound, which is our control, that's kind of, it's in the middle. That is very clear, that's very specific, and that's our healthiest oysters. So those are all very, very specific, but otherwise the Vibrios are broken into two separate areas in this tree. So that kind of tells us that there's definitely not just one species that is being opportunistic and harming the oysters. That means there's probably multiple, maybe 2 or 3, and that would definitely requires further research to look into what exactly those species are. For the NMR, the metabolites though, I want to talk to you about this all now. It's hard because there's not much research done on metabolites outside of human research. So basically what people have been doing so far in the scientific community of using NMR for oysters or fish or any other kind of thing that isn't human, they've been comparing it to the human data and doing it all by hand. So this looks really pretty right here, but if you printed it off, it could be like 7-8 pages long. And one peak doesn't correlate to just one metabolite, it could be 60 peaks. So basically until now, people have been using crayons and drawing which exact peaks associated with this exact metabolite. And that's painstaking work. So what I've been trying to do really hard this semester is to create a workflow in R using RNMR1D package to make that process so much easier. So hopefully next summer at the next mortality event, we can hit the ground running, get a bunch of samples, and be able to analyze them much faster than what's going on right now. So where to next? I'm really hopeful to get this workflow done soon, maybe by December, we'll see. But from that, I want to get a principal component analysis of the metabolites and be able to compare that to the results from the bacteria that we found. And I want to identify the specific Vibrio species, expand oyster sampling to wild diploid oysters. It would be really interesting to see how wild diploid oysters are comparing in survival rates to the cultured triploids. And if maybe there's different opportunistic bacteria, like, we don't know. It'd be really cool. But issues to resolve in the future, we definitely need to update our metabolomic sampling procedures because, as I was saying earlier, being able to find the metabolites gives you a snapshot of that exact time. So what we were doing was putting the oysters on ice and bringing them back to the lab without thinking that putting them on ice would cause a stress that would change the metabolites we see. So that's definitely something to resolve in the future. And we want to sample before, during, and after summer mortality events from here on out. But thank you so much to Dr. Ben-Hareen. He's the North Carolina State pathologist, shellfish pathologist, and he's the leader of the shellfish pathology lab at CMAST. He's been a huge help and taught me so much. And I also want to thank the NMR specialists, Maria Serrano and Dave Morgan, for teaching me everything I know about metabolites. All right, any questions? Thank you so much for listening.
Do you know if those Vibrio species are species that can be human pathogens?
I'm pretty sure they can. I remember reading this the other day, that it's just a matter of like how much is present, where it's like something like a little bit of Vibrio species can like affect oysters so much more than like human impact would be.
Cool, thank you.
Yeah.
Any other questions for Sophie?
All right, well, thank you so much for listening, everybody.
Absolutely.
And thank you, Dr. McKinney.
Yeah, great job. So if you can stop sharing your screen, perfect. Up next, we're going to have Kolby Altabet, a senior in fisheries, wildlife, and conservation biology.
All right, thank you so much, Erin. I'm going to start sharing my screen now. There we go. All right. So welcome, everybody. My independent project was on urban bird richness in Wake County. And I was looking at how different social and ecological factors might affect the bird biodiversity patterns across our county. I'd first like to take a land acknowledgement, and I hereby acknowledge the ancestral lands of the Tuscarora, the Catawba, the Siouan, which I'm currently presenting on today and conducted my fieldwork on. And I'd also like to acknowledge that the toll colonial took on the indigenous people, such as disease, displacement, and cultural erasure. I'd also like to do a quick research acknowledgement. I'd like to thank all the Triangle Bird Count volunteers who came out, the Terry Student Enrichment Fund, and the Office of Undergraduate Research for funding me, and everybody at the Lab of Reconciliation Ecology for helping me out. Dr. Kadi and the grad students, it was amazing. All right, so a lot of people think that cities are devoid of wildlife, but this can be further from the truth. All you have to do is look around and they're there. Here are some two examples. That is a red-shouldered hawk on the top, sitting in a construction site on Centennial Campus. And below it is a barred owl sitting in front of David Clark Labs. So the reason why cities are so complex is because they are coupled natural human systems. This means that both social and ecological factors combine together and influence one another. So social systematic structures like redlining, for example, can influence the ecology of a system, or on the vice versa, ecology can affect what happens in a human system environment. A good example of these two interconnecting is New Orleans and how Hurricane Katrina affected it. This is why when I was going for this project, I learned that citizen science does not equal community science. Citizen science relies on the recruitment of people who are naturally interested in scientists, but not the community members themselves. And this creates a bias in the observation, whereas community science relies on the specific recruitment of community members to do science in their own communities, which brings about environmental justice issues and ecological restoration. Which is where the triangle bird count comes in. This is a huge study done by Dr. Kolby and spearheaded by his grad students. This map over here was created by Michael Caslin, amazing GIS student. And what you see here is tons and tons of sites that I unfortunately could not get to all of them to create the perfect spatial pattern. But this is what we kind of aim to look at going forward to create better spatial coverage to understand every aspect of the social and the ecological workings. So my research question involved what are the definitive or maybe non-definitive relationships between avian species richness, between canopy cover percent, impervious surface percent, the type of land use, noise levels, and also a socioeconomic factor, which I used as mean housing prices of the neighborhoods. That was my current research that I did in the spring of 2019. I'm hoping later to maybe compare more of this to other cities. So comparing this to either from Raleigh to Fresno or Fresno, Phoenix, and Raleigh. We're really hoping to do some great work here. My objectives of the 2019 exploratory study was to understand if Wake County's widespread canopy coverage could be significant towards species richness across the urban-rural gradient, because as we know, this is the city of oaks. There's a lot of trees here in comparison to maybe some Western cities out west, like LA and Fresno and Phoenix that don't have that canopy coverage. We're also looking to determine if systematic structures of Wake County could explain these species richness patterns. So for my methods, let's just do a little line here. All the sites down here, these are all my sites. They include 50 randomly selected sites and 23 added sites that were done either by triangle bird count volunteers or the graduate students. And you can see that even with the random selection and just our slight recruitment that we had, we're still missing spatial coverage here, which is why the purpose of a systematic point count is to eventually get coverage over all of the years. Oh, let's see here. There we go. All right. So my methods include doing a 10-minute point count at each site. Ooh, hold on, let's see here if we can get those erased. There we go. All right. So my methods include you doing a 10-minute point count at all of our sites. And then before that 10-minute point count, at the 50 randomly selected sites, I waited 5 minutes before that to take all of my sound level meetings with a X-Text sound level meter. And for all the other sites, I took data from the National Transport Noise Map to get the noise level readings from that. For land cover, tree canopy coverage, and impervious surface, I got those from the National Land Cover Database. And for our socioeconomic factor, I took the mean housing prices from Gill estimates. There you go. All right, so when doing the exploratory results, I did a basic multivariable analysis to understand the different correlations that were happening between all my variables. And what I found were the two strongest ones that correlated with the number of species, which was the noise sound level right here that had a p-value of less than 0.001. Our next highest one was the number of species compared to the mean housing prices, which had a p-value of 0.0011. And then another thing that was very interesting to me was this one right here next to it, which is the species richness compared to canopy coverage, which almost has no correlational relationship. So to further look into why there isn't this relationship, I looked at the species richness and canopy coverage over different land uses, which was helping us understand the urban gradient of our sites. And as you can see here, they still don't have any really significant correlation. The developed high medium intensity one is almost there, but I think we would probably need more sites to fully understand if that's significant or not. And then next I did a fit least square to really hone in on those correlations. And again, we found the same things, that our noise level was very significant. The Zillow estimate of the neighborhood average was significant. And then also that the Zillow estimate combined with the average noise level was also significant as well. So after that, I did a stepwise modeling to see if those predictions were the same. And it showed us what we had been seeing, which was that the greatest predictor was the average surrounding noise level. Then the next one was the Zillow estimate neighborhood average. And then still significant with a strong p-value of 0.003 was our neighborhood average with combined of the surrounding noise levels, which kind of shows that our anthropogenic effects are really positive towards the species richness pattern. So because canopy coverage didn't have this effect on species richness like I thought it might have, I think this is due to the fact that canopy coverage over the entire Wake County land metrics is very spread out. And possibly because of this, this was able to reveal the stronger associated social systematic structures like the housing prices and the noise levels. And it was really interesting that the combination of the two also provided a really significant prediction as well. Because typically we think that, ecological factors are going to be the better predictors. But this just shows that we still have a long way to understanding the complexity of cities, and that we really need to maybe focus more on the community science and environmental justice to get the social aspects of what it is that complexities of cities have on biodiversity. In the future, I think it might be good going forward to understand the dynamics of the socioeconomic systematic structures of species richness, particularly redlining and species richness, and also the native plant composition / species richness compared with avian species richness across the socioeconomic gradient. And also, like I said in the beginning, understanding the differences between these socioecological factors between cities. So are cities inherently homogeneous with these factors, or are they heterogeneous? I definitely think it's something interesting going forward. But with that, I would like to conclude my presentation and open the floor for questions.
I have a question. Hey, Kolby, that was really awesome. The correlation between the species richness and sound is super strong. That's awesome. And I was wondering if you can remember any of the species that seem really tolerant to high noise. Do you remember which species those are?
Yes, actually, some of our most common species were cardinals and eastern mockingbirds. Those are the ones we typically saw even in sites that didn't typically have high richness, so like apartment complexes. Typically more of the generalist species were in those areas that had that, and even non-native species as well were typically found in those areas that had the higher noise levels.
Cool, thank you.
I might ask you a tougher question, Kolby.
Okay.
This is great. It's great to see these results and your talk. Thanks for doing all this. But one thing I'm wondering, and this might be something that we might get from a reviewer when we submit this for publication. How confident are you that the relationship between noise and number of species is not simply indicating that in noisy places you didn't hear them and you couldn't detect them?
Mm-hmm.
That it's a detectability issue rather than an actual difference.
Right. And I thought about that as I was looking at this correlation. I think it might be a combination of the fact that in noisy places, it's hard to detect things. And also, it might just be that it's a combination of there are lower birds and we can't hear them as well. But I think that especially going forward, it might be better to define what that detectability range is for the cities to understand the strength of this relationship.
But from your fieldwork, do you have a sense that you think you really missed birds in some places?
From my opinions, from my personal opinion, I don't feel like that. I think maybe that there might have been one or two that got caught up in like all the birds like singing at once, but I honestly don't think that detectability was an issue. It might have possibly been an issue for our volunteer samples because they're not ecologically trained as I was, and they were just given kind of like a step-by-step process as to how to do this.
Yeah, but also they are birders, and
right.
I suspect that most experienced birders will say what you just said, that they don't think they missed it because they have an ear for it. So yeah, okay, good job.
Thank you.
Do you have any other questions for Kolby? Awesome. Great job, Kolby. Thank you so much.
Thank you.
And if you could, yeah, emoticon reactions, welcome, for sure. And with this, I will introduce Rache Manning, a senior in zoology, who's going to wrap us up today.
Hello. I'm going to share my screen.
Looks great. I think you're ready.
We're all set. Okay. So hi, my name is Rache. My research this semester was centered around rock snot and what causes it. And so rock snot is formed by the diatom called Didymosphaenia geminata, or Didymo for short. And it is the algal bloom of this microscopic single-celled algae that is found in freshwater systems around the world. And though it's common in these freshwater systems, these blooms have only recently been documented in the past few years. And so when it blooms, it forms these stalks, which you can see here in the two pictures on the right. And when you have a lot of this stalk producing didymo, you can end up with this thick mat that kind of resembles wet toilet paper. So the diatoms attach themselves to rocks and then produce these stalks that sort of weave together into these wet, slimy looking clumps, which is why it's called rock snot. And what's interesting about didymo is that it tends to bloom in streams and rivers with very low levels of dissolved phosphorus, which is a little weird if you think of, or algal blooms, they normally are found in places with abnormally high nutrient levels, but didymo blooms are not common usually in waters above a specific phosphorus concentration. And so the big issue with rock snot is that since it's so dense and it grows into these very large mats, it can have really big impacts on the food web by smothering food sources for fish and even some abiotic aspects. It could possibly cause blockages in streams, and it could also cause economic issues where fishing is popular. And so what's unknown is what actually causes these didymo blooms. And the leading hypothesis is that the low phosphorus levels in these streams causes this diatom to produce these stalks. And so the objective for this research study was to measure and interpret the gene expression of different didymo samples and compare them to see how each of the didymo samples responds on a genetic level. And so we had a data set where stalk producing didymos was exposed to high phosphorus. And then the part of the research that I was more involved in was comparing blooming didymo samples or rock snot samples with non-bloom samples of didymo. And then there's also data for comparing stalk producing didymo that was transplanted into a stream where didymo usually does not bloom, but that is still in progress. So I have no results on that yet. And so the samples were taken in streams in Colorado, and you can see the dots on the map are sample sites. And the red sample sites are sites where bloom was present, and the yellow is where there were no blooms found. And so didymo covered rocks were taken from streams, and some of them were placed into tubs with a high concentration of phosphorus for a few hours, about approximately every 2 days. And then after 6 days, samples were taken of the didymo on the rocks and the didymo in the water to measure gene expression. And the lab methods, shotgun RNA sequencing was used to chop up the DNA pieces and sequence them to see which genes were differentially expressed between the compared variables. And so what I did was search the genes and the predicted proteins in a database to see what biological and molecular functions and pathways were expressed more or less in certain samples. And so for the samples that were exposed to high phosphorus, only 21 gene transcripts were differentially expressed, and none of them were associated with any diatoms or algae. So basically, we found no difference between the stalk-producing Didymo exposed to high phosphorus and the stalk-producing Didymo in its normal P, where it normally forms. And because there were so many genes that were expressed there and so little time in the semester, we have not yet finished processing the data for the bloom versus non-bloom samples. But so far, what we can see is that in non-bloom, there were a lot of bacterial genes expressed. And then in the bloom samples, there was a lot of viral genes and proteins expressed that we found. And the viruses that we found are known to infect marine diatoms. So I think that's pretty interesting that we could find viruses in these freshwater diatoms. And so this chart shows the up and down regulation of biological and molecular pathways. And basically what it's saying is that what was upregulated were functions dealing with lipids and RNA replication, which would help viruses because viruses have that lipid membrane and need to be replicated. And so what was downregulated were these pathways dealing with energy, like photosynthesis, and phosphorus pathways. And so basically, even though the data is not finished processing with what we have so far, I wonder why they produce these stalks. And so in this picture, you can see the little brown dots are diatom cells, and the stalks are the white. And so what the stalks are doing is they're pushing the cells upward. And so a hypothesis could be that they are trying to push them upward so that they could get more light, because sunlight is known to possibly kill viruses. And there's also exopolysaccharides in these stalks, which is known to have antiviral and antibacterial properties, which prevent viruses from attaching and replicating in cells. So that could be a reason why these stalks Didymo produce these stalks and happen to have viral genes expressed as well. And so I would like to thank my mentor, Dr. Brad Taylor, for allowing me to join him in his research and work with his diatom data this semester. I thought it was really interesting, and I'm just very thankful that I got to explore this topic more. And are there any questions?
I have a question. It's a little bit off topic, but I had never heard of rock snot before. I don't know what I would think that it was if I saw it in the wild. And I'm wondering,
Yeah.
like, have you talked to people who are like shocked by what this was? Or have you talked to people who've seen this in the wild and didn't know what it was before? And like, how do you explain it to them?
Unfortunately, I was not involved in the actual field methods, the data collection part. So I have never seen rock snot before, but I can imagine that people would be pretty confused. In what I've researched, it said that a lot of people maybe thought it was like sewage, which can also cause some big issues. No one wants to fish where there's sewage in the water.
Yeah, if it looks like wet toilet paper, for sure.
Yeah. Yeah. Not very appealing.
I'm just thinking free range kombucha SCOBY.
It does kind of look like SCOBY.
Very cross-disciplinary, right? Has anybody else got questions for Rache? Yeah, Dr. Leggett learned a new term today. Awesome. Well, thank you so much, Rache. And thanks to all of you who presented today. This is your reminder now. Let me give you the link. I've also been sending out reminders. Monday, we have part 2 on Monday from 3 to 4 p.m. So join us for the second installment. And in the meantime, if any of you are students and are now interested in the minor, I want to do that. I'm the person you talk to. So drop me a line. I would love to help you work your way toward achieving the minor. As our students today have shown. If you are faculty and say, I want to work with students, talk to me. I can help advertise your projects. So yeah, thank you so much to the 4 of you, to Jared, Sophie, Kolby, and to Rache for giving us a stunning insight to the diversity of research going on at the undergraduate level in our department and across departments. So yeah, thanks all. Thanks for coming. Happy Friday. So once again, thanks for tuning in to part 2 of the Applied Ecology Minor Symposium. And with that, we're going to start off with Emma Briggs, who is a senior in environmental science. So Emma, take it away.
Thank you. All right. Can everyone see that?
Yes.
Nice. Okay. Hi, everyone. My name is Emma Briggs, and my Applied Ecology minor project is on the efficacy of pan traps in estimating wild bee abundance. So wild bee species provide many essential pollination services in both natural and agricultural environments. And you can see an example of some of those awesome species on the image on the top right of the screen. However, pollinators across the globe have recently experienced an unprecedented decline in population. And so bee researchers are attempting to monitor these population changes through various different sampling methods. Pan traps are a super common passive sampling method for pollinator communities. And basically how it works is bowls that are painted with bright colors are filled with soapy water and left out in the environment. And so bees are attracted to the colors since they mistake them for flowers, and sometimes they fall into the bowls. And so the bees which fall into the bowls end up drowning, and then they are collected and processed for pinning and identification. However, there's been some recent research that indicates that pan traps may have some taxonomic biases, and that pan trap capture rates may not actually reflect the abundance of bees in a given environment. And so just to clarify, pan traps can't generate absolute population estimates, but researchers often use their capture rates to predict the size and community composition of bee populations. So in comparison, market capture studies are a less common active sampling method that can produce absolute population estimates. And I'll explain how those studies work a little later in the presentation. So the knowledge gap that I'm addressing with my project is that there are currently no studies that correlate absolute population estimates, such as those generated from market capture studies, with the capture rates of pan traps. So the main goal of my project is to determine the efficacy of pan traps through completing a market capture study. So my hypothesis for the project is that if pan trap capture rates are a valid indicator of bee abundance and community composition, then pan trap capture rates will be correlated to market capture population estimates. But we predict that these two measures will likely not be correlated due to suspected biases in pan trap efficacy. So the market capture study was conducted this summer at the Butner Falls of New Scamelands, which is right beside Falls Lake in Raleigh, North Carolina. We had 3 study sites, which were named Site A, Site B, and Site C, and you can see their locations on the top right of the screen. So the study utilized 3-day market capture periods for a total of 5 survey weeks. And each of these market capture periods were conducted every other week, so it took a total of 10 weeks to complete all of the fieldwork for the project. The target species that I captured were the eastern carpenter bee, Xylocopa virginica, the leafcutter bee, so we just narrowed this down to the genus Megachile, longhorn bees, Melisodes mimaculatus, and finally green sweat bees, which we narrowed down to 3 genera, which are in North Carolina, Agochlorella, Agochlora, and Agochloropsis. And you can see pictures of each of those species on the bottom right of the screen. So in addition to the market capture study, we used pan-trap and aerial netting data collected by the North Carolina Wildlife Resources Commission. So to complete the project, we actually piggybacked off of another multi-year study that was already ongoing through the WRC. So that way we only had to complete the market capture half of the experiment and not the pan-trap and the netting portion, since we could just use the data that was already being generated by the WRC. So the pan-trap and netting data wasn't collected by me, but it was from the exact same study sites and collected during alternate study weeks. So here's some images of each of the three sites. So they were generally located in forest clearings, and each of the sites had experienced a prescribed burn in the last 3 years. So the general procedure for the market capture study was that we first captured a bee on day 1 of the period, usually when it was visiting a flower. We then applied a unique daily paint color to its thorax and released it back into its environment. So the paint dot was applied in something called a bee squeezer, which is basically a syringe with a piece of netting at the top and a foam plunger at the bottom. So bees were kind of carefully herded into the tube and then squished up against the netting where we could paint their thorax. So on days 2 and 3 of the study, any previously marked bees were recaptured and given another unique daily paint color. And any newly captured bees were marked for the first time. So you can see a picture of a carpenter bee with a green marking at the top right of the slide, and a leafcutter bee with both blue and pink markings at the bottom right. So the daily number of captures versus recaptures was recorded and used in our statistical analyses. So to analyze our data, we first calculated absolute population estimates for each market capture period in a program called MARC. And once we had those estimates, we performed a linear regression between the market capture data and both the pan-trap and the aerial netting data from the Wildlife Resources Commission. And we did that in order to determine if pan-trap capture rates predicted absolute bee abundance. So we generated 22 market capture estimates out of 60 possible site and species combinations. So some of these estimates just couldn't be generated because of low catch rates or a lack of recaptured bees at the study sites. We needed at least one recapture to get the market analyses to work for each study period. So we took these market capture estimates that I generated and directly compared them to the pan-trap and netting capture rates that were recorded by the Wildlife Resources Commission. So the market capture estimates on these graphs are shown in pink, the pan-trap estimates in orange, and finally the net captures in black. And so just to pick out a couple of these graphs as an example, there were some species like the leafcutter bees, which seemed fairly well correlated between the market capture and the netting estimates, and others like the green sweat bees were less obviously correlated. And additionally, some species like the leafcutter bees or even the carpenter bees really didn't show up in the bowls at all, whereas the green sweat bees and the longhorn bees had fairly large capture rates. So this graph shows the linear regression between the pan-trap capture rates and the market capture population estimates. So we transformed the two axes in order to better see patterns in the data and to make sure our statistical analysis ran smoothly. And after looking at those previous individual species graphs, we decided that a week-to-week view of the data might be kind of problematic since it can hide broader trends that might be disrupted due to short-term weather patterns or other temporary environmental conditions. So for the next step of the data analysis, we pulled all the weeks together and then calculated the linear regression for all of the different sites and species. So each point on this graph represents a site and species combination. So for this regression, only 11% of the variation found was explained by the predictor, and the relationship between the pan-trap capture rates and the market capture estimates was not seen. So on the whole, the pan-trap capture rates and the market capture population estimates were not well correlated. So in comparison, the aerial netting capture rates and the market capture population estimates were well correlated, with 51% of the variation found in this regression being explained by the predictor, and the relationship between the aerial netting capture rates and the market capture estimates being significant. So inherently, this does kind of make sense since the market capture study was also completed using netting and capturing bees. So some of the challenges we faced in this experiment were low floral density. If there were no flowers blooming at the site, there were usually no bees present, which meant we either had imprecise or incalculable market capture estimates. Additionally, there were both weather and timing constraints for our study. So for example, we sometimes had to miss survey periods due to inclement weather. And the market capture and pan-trap data were also taken during alternate weeks. So the bee populations could have experienced changes due to different environmental conditions in the time between the two sampling methods. So possible solutions for these issues are a larger study area, which would hopefully always have some kind of blooming flower present. We could also try to implement a simultaneous pan trap and marker capture study, which would hopefully reduce any environmental differences across the different survey weeks. So the main takeaways from our results are that pan trap capture rates were not well correlated with bee abundance across sites and species, whereas the netting capture rates were well correlated. So this is what we initially predicted in our hypothesis, which is really great. We also wanted to note that we observed really low pan trap capture rates across all of our study species this summer, especially in comparison to pan trap data from previous years at the exact same study sites. So we concluded that in this particular environment and in this particular year, pan traps yielded a low number of bee captures. But pan traps may end up having better capture rates in different geographic areas or even different sampling years. So the results of this project have some useful applications. Hopefully the study will increase awareness of the potential limitations of pan trap data and inform researchers about how pan trap capture rates may not accurately quantify absolute bee abundance. However, our data does indicate that pan traps can still be useful in sampling bee species that are often net shy. So for example, the longhorn bees in our studies were much more often captured in the pan traps than in aerial netting. So pan traps can still be a useful tool in some cases, despite their quantitative shortcomings. In conclusion, pan traps are limited in their ability to estimate absolute abundance of bees, but can still be really helpful in determining if a certain bee species is present at a particular. So I would just like to thank my awesome mentor, Dr. Elsa Youngstead, for making this project possible, and also Dr. Jaime Collazo, who was super helpful in guiding us through the MARC program software. Any questions?
Awesome. Thanks so much, Emma. I'd encourage all viewers to make use of your reaction emoticons if you want to applaud silently, or feel free to unmute and ask questions.
Hi, Emma. This is Jim Rice. Great job. Excellent seminar.
Thank you.
I was curious, on average, kind of how many bees you caught with each method during a particular survey. I don't do local conversions in my head very well.
Each day and like each study site, we would get anywhere from like 20 to 40 bees probably. So yeah, our total was like 500 something bees over all of that. So we had pretty good netting capture rates. But then the pan trap data, we only got like maybe 10 to 20 captures for like all of the species in the pan trap. So we had really low numbers this year for some reason.
Oh, okay.
Mm-hmm.
Hi, this is Becky. I enjoyed your seminar. That was great. I was wondering if you had any speculation for why your pan traps yielded so few bees this year.
I was thinking about this a little bit. So we had certain, like during some of the survey windows, the weather was incredibly hot, like upper 90s every single survey day. And so I just noticed walking around netting bees that there weren't many out. And so I do wonder if weather had some impact. Maybe this summer was really hot and that impacted our number of bees in the bowls.
Thank you.
Anybody else? One final question. All right, well, thanks again, Emma. And up next, we've got Sydney Kuczenski, who is a sophomore in marine science. So Sydney, you should be, there we go. Yeah. Perfect.
Hey guys, hi, I'm Sydney Kuczenski, like Dr. McKinney just said. My study was a fisheyes view, studying color perspectives. Well, let's see if I can get it to move. All right, so in particular, I looked at goldfish at a aquarium that I interned at over the summer. And just a little background about goldfish, because it's probably a species you haven't really thought much about. But goldfish are actually descended from the Asian carp kochi. They're generally a more grayish dual color fish. So every so often, a mutation would come along that would cause the fish to have gold coloring, and those goldfish were preserved and shown mercy rather than eating them. The people that bred these Asian carp thought, wow, this is really cool. We're gonna save these, goldfish. So over time, goldfish have become a pet. There's actually a bunch of different breeds, including fancy goldfish, fantail goldfish, lionhead goldfish. I could go on and on. Originally, these Asian carp / goldfish were from Asia. So outside of Asia, goldfish are actually an invasive species. So my research question for this study was, what colors will affect aquarium goldfish and or visibly stress them out? So this was an observational study, so I could really only test visible stress of the goldfish. And people ask, what signs of visible stress could I actually test? And usually that would be seeing the fish darting around, hiding behind decorations or in plants, just panic swimming around and around the tank, or sometimes even not eating if they got so very stressed. I'm going to go ahead and do the applications of this study. So the internship that I had was at the Catawba Science Center in Hickory, North Carolina. And this study related directly to the aquarium because we were remodeling this summer due to COVID-19. We weren't open. So we were doing a bunch of new habitat design, and my mentors wanted to look at what colors will kind of stress the fish out more, or, you know, what makes them comfortable in their captive environments. Another application of this study would be in fishing. As you go deeper in water, different light is available, or different light waves penetrate through the water. So deep sea fishermen need different lures than offshore fishermen. So knowing what light fish can actually see can help industry fishing. And finally, goldfish actually see about the same wavelengths as humans, which is really interesting. Goldfish are often used in optometry studies for humans. So goldfish can see red, green, blue, and UV light waves. Humans can only see red, green, and blue. You might ask, why does a goldfish see red, green, and blue light waves? And it's generally so they can hunt. So goldfish use their sight as well as their lateral lines and sense of smell to search out food, hunt for food in the water. But because goldfish can see in the UV wavelength spectrum, and UV light penetrates water so much better than the visible light, they generally use that aspect of their sight more than they use the red, green, and blue visible light. So my hypothesis for the experiment was that warm colors, red and yellow in this case, will stress out the aquarium goldfish because there are colors that they're not often exposed to. Cool colors, however, like blue and green, will not stress out the aquarium goldfish because they are colors that they are often exposed to. So here's a few pictures of my experimental design. On the left, you can see the tank before we've done anything for the experimental design. The goldfish's names were Lilo and Stitch. We just named them. We have a fun time naming them at the aquarium. On the right, you can see my method for holding the colored paper that would provide the experimental side of the tank. Here's you see the actual experimental setup. So we would take pieces of colored paper, red, yellow, green, and blue for the experiment, and we would cover half the tank, providing an experimental side of the tank and a control side of the tank where there would be no colored paper. In this picture, it looks a little dark on the experimental side of the tank. That's just the way I had to get the pictures set up just to get good exposure. But in actuality, there was equal amount of light on both sides of the tank. So for my data collection, what I would do is every hour for 3 days, I would stand a few feet back from the tank and look and see where the fish were, do a point summary. And generally, if I didn't see them, it was clear they were on the colored side of the tank. If I did see them, they were obviously on the control side of the tank. If they were in the middle, just for simplicity, we considered them to be on the colored side of the tank. We did this every hour for 3 days, 3 consecutive work days, so about 7 to 8 hours apiece. And then the fish had 4 consecutive days off to not stress them out too much more. So individual results, you see Lilo on the left and Stitch on the right. Both of them seem to like blue the best. As you can see, they spent more time in the blue side of the tank than the blank side of the tank. That's the bars right at the top. They seem to like green the second best, then yellow, then red. Together, this was just taking their overall average across the 4 weeks for each color trial. Again, you can see they like blue the best, then green, then yellow, then red. So the results of this study clearly showed that my hypothesis was correct in assuming that warmer colors will visibly stress them out, make the fish stay on the control or blank side of the tank more often than they were on the colored side. That's kind of what this graphic is showing. They don't like the yellow and red, they do like the blue and green. In the future, I would like to have more fish and also try to test a UV light and see if that stresses them out or if they like that better. Finally, I would like to thank the Catawba Science Center for the use of their facilities, as well as Molly Jordan and Casey Farthing for their mentorship throughout this project. Any questions?
Thanks, Sydney. Has anybody got questions?
Hi, this is Becky. I enjoyed your seminar. What color were the tanks prior to remodeling them? Were they the blue colors or the red colors?
Prior to remodeling them, we had mostly just rocks in there that were pretty neutral colors. Afterwards, we added some plants. We've also taken our inventory of plants that had green and red and purple and all these bright colors that you normally wouldn't find in natural environments and changed it to reflect the study's results. So green, blue, sort of those cool colors that you would find in a fish's natural environment.
Thank you.
So I've got a question, Sydney. It looks like neither Lilo nor Stitch liked those bright, warm colors. Have you noticed any avoidance behaviors? Like if there's a pack of kids, you know, sometimes school groups, I mean, pre-COVID would all come in wearing the same neon colored shirts, so they're easy to find. Do you predict that that might really stress out your fish populations or?
Yeah, so that's a really good question. Generally, as the length increases between a fish and a color, they can't see that as well. So just like how depth, light doesn't penetrate as far, distance, it doesn't either. So if a fish is on the inside of the tank and there's a glass wall between them and the pack of kids, they're probably not going to really notice them. More than anything, they might notice the noise and of just the kids talking and yelling and screaming. But fun tip, if you want zoos or aquarium animals to come up to you, wear the same color as the zookeepers. We wear khaki, which is often a neutral color, doesn't really stress any animals out. But if visitors started wearing khakis, the animals have associated that color with food, so they would come up to you. But just a fun fact.
Pro tip. And that's the opposite advice I've heard of: never dress like a veterinarian, because then you get total avoidance. Thanks again, Sydney. If you can stop sharing your screen, then we will wrap up with Nick Moore, a senior in environmental science. And Nick, just remember to unmute.
Got it. All right. Let's see. Ready when you are.
Perfect. Good to go.
All right. So my name is Nick Moore, and the project that I worked on this semester is titled Exploring How a Sunflower Pollen Diet Can Impact Gut Parasitic Load in the Common Eastern Bumblebee. So the focus of my study was on Bombus impatiens, and these bumblebees are very efficient pollinators for both fruits and vegetables. They visit more than 90% of the top 107 global crops, and some of these might be like apple trees, raspberries, tomatoes, blueberries, pretty much anything you can think of. And the species is also mostly found on the eastern side of the US, although some people have moved them around. And they're in pretty much any type of habitat, ranging from a temperate forest to like urban cities. So we know that bumblebees are largely on the decline due to anthropogenic effects, but the problem at hand that we looked on for our study is a protozoan gut pathogen that's called Cryphidia bombi. And these gut pathogens are harmful to bumblebees because they can reduce their motor skills, they can reduce their foraging potential, and their overall colony reproductive success. So the pathogens can be contracted by consuming fecal matter from other bees orally, and this usually happens at like a shared flower site or within their colony hives. And you can see from the GIF at the bottom that the poop is directly landing on a flower, which is a problem. So here's some background info on sunflower pollen and how it's capable of reducing pathogen loads in bumblebees. Sunflower pollen has shown effects of increasing microcolony reproduction, as well as having properties that make it antimicrobial. So we were curious why sunflower pollen has these effects and if it's related to like its physical or chemical makeup. And as you can see on the left, there's a figure that compares the load of Cryphidia found in bumblebees after they were fed different diets. And it's noticeable that the sunflower diet definitely resulted in a far less Cryphidia cell count when you compare it to something like a buckwheat or rapeseed diet. So our first objective, we wanted to have a better understanding of the mechanism that might drive medicinal effects for reducing gut pathogens. The image on the left is kind of a microscopic view of sunflower pollen, and it shows physical properties that make it look like a spiky ball. So this might help expel pathogens by sort of acting like a laxative with its texture. And then on the right is lauric acid, and that's a fatty acid that's found in sunflower pollen. It's also found in coconut oil, and that can help humans with like yeast infections and gut pathogens. So it's unsure as to which one of these factors or both of them might aid bumblebees in reducing Cryphidia pathogen loads. And then going more narrowly on the objective, we wanted to test the individual and combined effects of a pollen diet, specifically wildflower and sunflower, and Cryphidia infection on the volume of feces deposited and the gut transit rate of fecal deposits. So our hypotheses, one, we thought the volume of feces deposited would be significantly impacted by consuming sunflower pollen diet. Two, that the rate would be significantly impacted by consuming sunflower. And three, that the infection by Cryphidia would significantly impact the rate of bumblebee fecal deposits. And I thought personally that the sunflower would make the bees poop more and at a faster rate. And I thought that Cryphidia would make the bees also poop at a faster rate. So getting into the methods, I wasn't around for the setup of the experiment due to COVID restrictions and not being able to work in labs. So instead, my job is primarily through image and video analysis. Before I arrived though, Jonathan, who's an NSF graduate research fellow, set up this 2x2 factorial experiment where bumblebees were fed either a sunflower or a wildflower diet, and then they're either infected or uninfected with Cryphidia. And then all of the bees were given pollen diets that were spiked with like a red dye, and that helped us better visualize their poop deposits. So on the right, you can see our experiment setup, and we had the bees in plastic containers, and they would sit on a paper disc, and then all of that would be on a glass table, and underneath the glass table, we had a camera set up to take photos and videos. And this is where I really come into play with analyzing data online. So initially, I just looked at the photos from all the bee samples, and I analyzed them in a program called ImageJ. And ImageJ allowed me to distinguish the count of poop events as well as their area of fecal deposits for each paper plate. And then it picked up anything that was about 5 millimeters squared or bigger, and it would label it red and have like a cyan outline. And then for statistical analysis, we processed images from 5 rounds. Each one had about 24 bee samples, which result in like 120 bee images total. And our response variables were the area of feces as well as the count of deposits. And our model parameter interactions that we tested were their pollen diet and infection treatment. And then we also include some covariates, which were pollen consumption, nectar consumption, and parasite load. And then with all the data, we conducted a general linear model regression and then a Shapiro-Wilks distribution test. So eventually the test led us to creating this bar graph on the right, and it shows how sunflower pollen actually had a much larger increase in the volume of fecal deposits as compared to a wildflower diet. So the y-axis shows the area coverage of poop deposits, and as you can see, the deposit was almost doubled when you compare it sunflower to wildflower. And the left side of the page kind of visualizes this difference in the bar graph. So the area of the sunflower pollen is definitely much larger than that of the wildflower. And then here we have a similar graph. This one, though, shows the total number of fecal deposit on average. So sunflower ranged about 6.8 deposits per bee, whereas the wildflower diet was more like 4.2 deposits. So currently my prediction is holding true that that sunflower pollen diet does result in a higher volume of fecal deposit. And then just as a side note, Crithidia did not alter the volume or number of fecal deposits. And then moving on to the second part of the methods that I was in charge of was the video analysis. So here we have a picture that's kind of the experimental setup with the camera placed underneath the glass, and it views all of the samples within one round. And my job was to watch most of that footage and find the timing of the first 5 red poops per enclosure. And I also had to record the number of red deposits in images, and I would screenshot those at different hourly intervals. So this is what it looks like at the moment that a bee poops. It kind of starts off black and then it fades out, and eventually it kind of transitions into like a red color. And then this is what it looked like when I compared the number of fecal deposits only by looking at images at differently hourly intervals. And as the video progresses, you can see that there's more red deposits that appear. So like at hour 1, there's only 1 deposit, but then at hour 13, there's like 6 that appear. And then for the video statistical analysis, we analyzed videos from 5 rounds again. These were each 15 hours, which totaled to about 93 total hours of footage. And then we did a GLM statistical analysis for rate analysis, and then a Cox proportional hazards model for time to event analysis. And then we use the same model parameter interactions as we did before with the images. And then this one, it's the time to event test. It's kind of complicated to look at in a short period of time. So I'm just going to show that if you look at the red dotted spots, those show healthy bees that weren't infected with Crithidia and they were fed sunflower. And they had a higher probability of pooping their first red deposit earlier than all the other interactions. So the healthy sunflower combinations pooped earlier than the other treatments as a trend. This one's a little more direct, and this one shows that the Crithidia infection increased gut transit overall. So the last one was just the first poop, but this is for overall. And neither of the pollen diets affected gut transit rate overall. The y-axis shows that poops per 2 hours, and regardless if it was a sunflower or a wildflower diet, it was roughly 0.3 poops per 2 hours when they were uninfected, whereas when they were infected, it was more like 0.5 poops per 2 hours. So just to recap everything, do the bees have a greater volume of fecal deposit when consuming sunflower? Yes. Did they poop at a faster rate with sunflower diet than wildflower? Yes, but only for their first like poop event, not overall. And then did Crithidia increase the rate of feces deposited? Yes. So before I jump into the discussion, there were some challenges that we approached. And one of it was that the bees would burrow holes into the paper, and that caused the image data to be harder to analyze. So to fix this, I had to take the raw image into paint and then use a red spray paint to fill in any holes where there would have been a fecal deposit. So that way the program would register it. And then also there were some very rare situations when it was just kind of hard to deposit or to distinguish new deposits in videos. And one time I noticed during a video that somebody closed a loud door and 2 bees pooped at the exact same time. I don't know if that was just coincidental, but I thought it was strange. And then we had to eliminate 1 round from our statistics because we used different methods for it. So with our data, we might be able to see some bigger picture. We know already that sunflower pollen can reduce gut pathogenic loads. And from our study, we might have more evidence that demonstrates how sunflower properties could lead to more pathogens being expelled from the body. Also, if humans utilize healthier pollen diets, such as a sunflower diet or a mix of sunflower, it could help revitalize bumblebee communities and promote greater pollination of our agricultural crops. So just to close things out, I want to mention some directions I think the study can go. One, I think that we should observe the fecal deposit rates and loads with other pollen diets, specifically other sunflower species. And I think we could study further into the mechanism that causes bumblebees to increase their deposits, whether it be mechanical or chemical. And finally, we should test how sunflower pollen diet can be used, or like how the sunflower diet can be used on other species, such as like moths or beetles. So yeah, big thanks to my mentors, Jonathan Giacomini and Rebecca Irwin. They were amazing. They helped me so easily with all the methods and everything, and they always got me on track, and they're just amazing. So thank you guys. But if you guys have any questions, feel free to let me know.
Thanks so much, Nick. Has anybody got a question? Yes, I'm thinking about the logistics of maintenance, right? So in your discussion, you talked about increasing maybe the availability or the use of sunflowers to help manage and clear some of those infections, right?
Mm-hmm.
But you also talked in your introduction about how the fecal deposits themselves can be a mode of transmission.
Mm-hmm.
So how do you figure, is it, I imagine it's not immediate, right? It's not an immediate laxative effect of like a bee consumes sunflower pollen and then immediately poops. So how do you, yeah, I'm just thinking through the logistical challenges of that, right?
I did talk about that the other day with Jonathan. He like mentioned, you know, they might help them, but then they're going to poop more. So then it might be easier to spread. But we don't know if the parasite's still going to be like as effective after it's been pooped out one time. So that's a whole new question that we could look at.
Gotcha. Yeah, if it's not resilient to environmental exposure. That's a really cool thought. Thanks, Nick.
Mm-hmm.
Anybody else have a question?
Yeah, I do. So where is this infection usually found? Like, is it a North Carolina thing? Is it North America? Or
I don't know if it's regionally specific. I haven't actually thought about that, but since the bees are kind of just roughly found around the East Coast, I think it can be anywhere. I think it's just like a very common pathogen for them. Any other questions?
I'm doing my long online pause to make sure I don't stomp out anybody else's thought process.
You may have talked about this a little bit in your introduction, but how do you think this is working? Like, what is it about sunflower pollen that's accelerating and increasing the rate of pooping?
So yeah, that's like the big question. I think personally that it might be something more to do with the acid compound chemical makeup of the sunflower diet, just because there's all the evidence of coconut oil having similar chemical properties, and it helps humans with our own clearing our system with gut pathogens and stuff. So I feel like chemical might be more important than its physical makeup.
Thank you, and great talk.
Thanks.
Final questions from anybody, for anybody? Well, this is the point where I'm going to say thank you again to all of our presenters. Y'all did awesome. And thanks to everyone who joined us. If you are a student interested in pursuing the minor, talk to me. If you are a faculty member interested in working with students, talk to me. Yeah, thank y'all so much.